Hello, budding biologists. Have you ever wondered how the information encoded in your DNA is used to make all the proteins that make you, well, you? It's all thanks to the process called transcription. Imagine it like copying a page of notes from a textbook into your own notebook. The cell does the same. It copies genetic information from DNA into RNA. This process is crucial for making proteins, which are responsible for nearly every function in your body. In fact, without transcription, your body wouldn't be able to perform even the simplest tasks. Let's dive into how this magical process works. Now, let's zoom in on the DNA itself, the place where transcription happens. The DNA that gets transcribed forms what we call a transcription unit. This unit has three important regions: the promoter, the structural gene, and the terminator. Think of the promoter as a start signal, like a green light telling the RNA polymerase to begin copying. The structural gene is like the page of notes that gets copied, and the terminator is like the stop signal telling the polymerase when to stop copying. In this carefully orchestrated process, only one strand of DNA is used as the template for creating the RNA. Isn't that fascinating? Here's where things get really interesting. The enzyme RNA polymerase is the workhorse of transcription. It binds to the promoter, and using the DNA strand as a template, it begins building a strand of RNA. This happens by following the rules of complementary base pairing. So, where there's an A in the DNA, RNA will add AU instead of the T you'd expect in DNA. The polymerase moves along the DNA, adding nucleotides to the growing RNA strand. It's like a tiny construction worker building the RNA one piece at a time. As the RNA polymerase moves, it opens up the DNA double helix and adds RNA nucleotides in a sequence that's complementary to the DNA template. Pretty incredible, right? Now, you might be wondering, why doesn't the cell use both strands of DNA as templates? That would be like writing two different versions of the same recipe, which could end up with confusion. If both DNA strands were copied, you'd end up with two different RNA sequences, and that could mean two different proteins, which is not what the cell wants. So, to keep things simple and organized, only one strand is copied, ensuring a smooth, orderly process of transcription. Once the transcription is complete, the result is RNA, but did you know there are different types? In bacteria, there are three main types: mRNA, messenger RNA, tRNA, transfer RNA, and rRNA, ribosomal RNA. mRNA carries the instructions for making proteins from the DNA to the ribosome, tRNA helps bring the right amino acids to build proteins, and rRNA is a key part of the ribosome, where proteins are made. Isn't it amazing how all these different types of RNA work together to make proteins, the building blocks of life? Let's compare how transcription works in bacteria and eukaryotes, like our own cells. In bacteria, transcription happens in the cytoplasm where the DNA is, and it's fast. As soon as the mRNA is made, it can be translated into protein. But in eukaryotes, things are a bit more complex. Transcription takes place in the nucleus, and the RNA produced is then processed before it can leave the nucleus and be translated into protein in the cytoplasm. This processing includes splicing, where introns, non-coding regions, are removed and exons, coding regions, are joined together. This extra step adds another layer of regulation and ensures only the right parts of the gene are used in protein synthesis. Now, let's dig deeper into how eukaryotic transcription works. Eukaryotic genes are a bit more complicated because they have both introns and exons. After the RNA is made, the introns are spliced out, and only the exons are left to form the final mRNA. But wait, there's more. The mRNA also gets a cap on the 5 end and a poly A tail on the 3 end. These additions help protect the mRNA and help it get exported out of the nucleus. Isn't it incredible that cells have these extra safeguards to ensure the mRNA is ready for translation? It's like packing a suitcase carefully for a trip. Everything must be in order before you leave. Let's wrap up our discussion on transcription. So, what exactly happens in transcription? It's the process where genetic information from DNA is copied into RNA. This RNA acts as a template for protein synthesis, guiding the cell in building the proteins needed for various functions. To help you relate, think of transcription like copying a recipe from a cookbook. The recipe, just like the genetic code, provides instructions that help us make a dish, or in this case, a protein. Without transcription, we wouldn't be able to make those important proteins. Now, the key player in this process is RNA polymerase. This enzyme is responsible for reading the DNA template and synthesizing the RNA molecule. It's like the chef who follows the recipe and prepares the dish. Finally, transcription occurs in the 5 to 3 direction, ensuring that the RNA is synthesized in the right sequence to match the DNA code. This directionality is crucial for accurate genetic information transfer.